Hi, my name is James Fry, and this is my gut microbiome presentation. A gut microbiome is types and proportions of microorganisms living in a given location in a animal or bacteria. The gut microbiome in animals is 40% Firmicutes, 30% Bacteroidetes, 10% Actinobacteria, and the rest of the 20% is spread among other little bacteria. The gut microbiome has been shown to be correlated to diet, environment, and other health aspects. A healthy gut microbiome, it can result in weight loss compared to an unhealthy gut microbiome. We questioned in this experiment, can diet affect a bird species gut microbiome? And our hypothesis is the gut microbiome of two different bird species is has similar compositions of bacteria, but due to their diet will have different amounts of that bacteria. In this experiment, we compared a 6-month-old 1-pound male pigeon to a two-year-old 2.5-pound female chicken. Initially, we compared the first pigeon to another pigeon, but due to a mishap, we had to compare to a chicken instead. For the DNA of for the pigeon and the chicken, to isolate it, we use the Qui-Gon kit. And to concentrate that DNA, we use the Zymo kit. The UV spectrophotometry was used to quantify the DNA so we could find the concentrations and the 260/280 ratio. We amplified the DNA using the Amplicon PCR. Specifically, we amplified the 16S rRNA gene, regions V3 and V4. The target DNA was first denatured at 95 degrees Celsius, and then it was cooled, and then we heated it up again to 72 degrees Celsius, so the DNA polymerase could bind and start synthesizing the DNA. This was cycled 25 times to make sure we had enough DNA. We used the gel electrophoresis to check the products of our PCR reactions, and then we used the Agen Ampere XP kit for the PCR cleanup. Indexing the PCR had 8 cycles of 95 degrees Celsius, 55 degrees Celsius, 72 degrees Celsius for 30 seconds each. And then after that, we reheated it at 72 degrees Celsius for 5 minutes. We then determined the nucleotide sequence of the 16S rRNA gene and added a unique set of short tags at the end of it. After that, we did the PCR cleanup again to remove old unused primers, and then we quantified the DNA again, but this time we used the fluorometric DNA quantitation because it is more precise than the gel electrophoresis. For our UV absorbance spectrophotometry results, sample 348 had a concentration of 25.4 nanograms over microliters and a 260/280 ratio of 1.77. Our other sample, which ended up not using in the end, sample 538 had a concentration of 0.8 nanograms over microliters and a 260/280 ratio of 1.77. So both samples were still pure because they were in the 1.5 to 2.2 ratio. Sample 538 did not have too much DNA, but still enough. And our sample 348 had a good amount of DNA in it. So again, our readings never came back, so we had to use past readings of sample 348 and a new sample of a chicken, sample 880. And for the kingdom, both had a lot of bacteria in them, but the sample 348, the pigeon, had some unknown and some archaea in the kingdom also. For order, both had consistent clostridiales, but sample 880 had a lot of bacteriodiales, and then sample 348, the pigeon, had more unknown and sphingobacteriales. For genus, both had a lot of unclassified. Sample 348 had more Phingobacterium, and 880 had more bacterioids. Of course, unclassified means that this sample was too small to actually get a read on it. For sample 348, there was plenty of DNA in there, and it was in the 1.5 to 2.2 range, so it was still pure. For sample 538, there wasn't too much DNA in there, but it still had a concentration, it still had, it was still in the 1.5 to 2.2 range, so it was also pure. The reactions for the jet electrophoresis did work as expected. Positive the positive control and sample 348 showed a lot of DNA. Sample 538 did not show too much DNA, but there was still DNA in there, and the negative control showed no DNA whatsoever. For sample 348, there was a high concentration in there, enough for sequencing, and for sample 538, there was also a high concentration in there to be able to sequence. The positive readings showed that the E. coli only had DNA in it, and the samples having the same bacteria in the kingdom meant that there was cross-contamination that occurred. Since we had to get past results and we couldn't use our samples that we experimented with, we did have enough reads for analysis. For the most part, the kingdom, order, and genus were relatively the same in the chicken and the pigeon. However, the chicken had a larger bacterioid genus, and the pigeon had a higher Sphingobacterium genus. Also, the chicken had a higher bacteriodale order. And knowing this, we can conclude that we can support our hypothesis that the diet does influence the amount of bacterium in gut microbiomes. Thank you very much.